5 o'clock, and we're happy to start the 2023 series of a Talk in 3 hour webinar. And my name is Georgia Parlocca. I'm hosting together with Silke Keller. And we're really happy today to speak about in situ monitoring of 3D cell cultures. And today we have a tandem presentation, so 2 speakers. And I'll let Silke to introduce them.
Yeah, thank you, Georgia, and welcome also from my side. First of all, thanks to our speakers. I'm really happy to have the both of you and to give our audience a little introduction. So I would like to start with Julia. Julia studied pharmacy at the University of Freiburg in Germany and received her approbation in 2014. In May 2015, she joined the Schenke-Leyland lab at the University of Tübingen, also in Germany, for her PhD, in which she was working on imaging in the field of tissue engineering to investigate cryopreservation of heart valves using Raman spectroscopy to detect ECM damages. And after her PhD, Julia became the group leader for Raman imaging and microspectroscopy in the Schenke-Leyland lab. And since last year, she also runs her own group at the NMI, that's the Natural and Medical Science Institute in Reutlingen, also in Germany, where she and her group develops imaging techniques for biomedical applications. And then as second speaker, we have Torsten Mayr. Torsten received his PhD in chemistry from the University of Regensburg in 2004. From 2002 till 2000, I'm sorry, that was wrong with the year, but from 2002 to 2004, he was a postdoc fellow at the Karolinska Institute in Stockholm in Sweden. And then in 2004, he became an assistant professor at the Institute of Analytical Chemistry and Food Chemistry at the Graz University of Technology in Austria. And then in 2014, he became an associate professor at the same institute, where his research is dedicated to optical chemical sensors and their application in biotechnology and environmental analysis. Between 2017 and 2019, he was a member of the editorial advisory board of the ACS journal Analytical Chemistry. And since 2017, he also became the CEO of PyroScience AT GmbH in Graz, which is a subsidiary of the PyroScience GmbH in Aachen in Germany. And since 2020, he is also a board member of the EUROX, that's the European Society for Organ on Chip. And we were happy to have the both of you there. And to not take away too much time, I would hand back to Georgia, who quickly goes over the housekeeping rules. And in the meantime, I would already start a poll that our audience is welcome to answer.
Yeah, well, we have few rules. First of all, mostly for the discussion, we would like to ask you to use the Q&A function that is in your Zoom app. And we will use the chat mostly to send you links and also information about next appointment. We would also like to inform you that you can request a certificate of attendance, and we will also give you some instruction in the chat, and that all of our webinars are recorded, and you can stream them and also rewatch them as many times as you like. And again, you will receive everything in the chat, or you can contact us.
Okay, yeah, I'm very happy to present here the technology that we are developing here at the Institute of Analytical Chemistry and Food Chemistry here at the Graz University of Technology. And I'm trained chemist, so I'm not so into cell cultures and this, but I'm really cooperating a lot with groups using that techniques. And yeah, if you look at the polls, so we have a lot of CD, we have the majority do 3D cell culture and also 3D culture, and a few people do also organ and chip and organoids. And I think what I will show today is suitable for 2D cell cultures or for all of these areas, especially and I will also show things that you can use for organ on chip and organoids. So yeah, I think the new year started with really great news, fantastic news, and it was obvious that the FDA, the new medicines, does not have not to be tested with animals anymore. So there are other methods are suitable, and I think this was also legislation signed by President Joe Biden. So I think this was really fantastic news for the whole community, especially here in this audience, in this 3R audience. So, but it was an article in Science about that, and this also states that this will not happen that fast, and there's a reason. Of course, technology is not so mature as organ-on-chip and organoids, and also others, but there will be some time. And one reason is also that we always have to want to know, and especially for me as an analytical chemist, what's inside these microfluidic chips or these microfluidics. So, and to analyze these beautiful biology, cell cultures, it's really a challenge. And there are not a lot of technologies available. So when I want to look inside, and so I can use sensors, and that's what I'm working on. So we really want to provide inline sensing. So that means the sensors are integrated directly at the cells. And why are these needed in microphysiological systems? So what you can do, you can use it for two measurements. One is, of course, monitoring and control of your cell culture conditions. That is very useful, and it's very necessary if you want to have really reproducible cell culture systems, especially in the microscale. And the other possibility is measure with these sensors the viability and the metabolic state of the cells. And this delivers you then metabolic data for disease and toxicity models. This can be very, very useful. So what is going on inside an organ-on-chip? So usually what you use to analyze these organ-on-chips or microphysiological systems is, now I come back to our poll. You say you use microscopy techniques or biochemical assays or immunocytochemistry. Okay, that fits to what I'm think is usually currently readout is dominated by light microscopy, also staining and fluorescence microscopy. So, and mainly fluorescent assays and light microscopy. So the problem with that is that this is usually qualitative, it's non-automated, and these are endpoint assays. So you cannot make a continuous measurement with them. And usually you don't get a measurement of the relevant metabolites. So what we do, we integrate small sensor dots into these systems. And with these systems, we enable non-invasive measurements. So and it's really miniaturized in close proximity of the cells. We can enable a contactless readout, and they are really in situ measurements and in real time, apart from other benefits that are listed here. So, and how does this work? So what is an optical chemical sensor? You see here in this sketch. So, and here the main part is a luminescent sensor spot. So, and this sensor spot is a host polymer where an indicator dye is embedded, which then interacts with the analyte or with the substance you want to detect. And this is then spotted or immobilized, this host polymer on the wall of your microfluidic chip or of your cell chamber, and then read out via light guide, where you can transport the excitation light and also the emitted light. So that is how the general principle behind the the optical chemical sensor. And you see here, this is how it looks like in reality. You have this microfluidic cell chamber, and where you can grow the cells. And here are two integrated sensor spots. They are in the range of 700 micrometers. So, and for this, then you can read out from contactless to the general bonds with this optical fibers. And what we can do now, apart from monitoring control the cell culture conditions, is really respiration and acidification measurements. Maybe a few of you know the seahorse system from where you also have this microtiter plate format, where you also measure oxygen and pH, and then make these oxygen rates and also the acidification rates. So we made a first model system, or first system with very simple chips. Also, they had been also prototype chips, and we used made a new type of prototyping. We tried out here. That's why it looks a little bit strange, maybe for some people, or not that nice. But it worked perfectly because we had a 6.5 microliter volume, and we used human lung carcinoma cells, which were grown in these cell chambers. And you also see here then these miniaturized sensing spots. So, and what we do now, we have, after seeding the cells and after incubation, we have a constant flow of 10 microliter. And then when you see that in here, this is the oxygen and this is the pH. And you see here, this is the flat line here. And as soon as you stop the flow, you see the respiration measurement. So you see how the oxygen is consumed by the cells. And also how the cells produce protons and CO2, and this is also resulting in a decrease of the pH. So from that measurement, we take 2 minutes, and in these 2 minutes, we calculate the rate. And if you then expose the cells to in this here, it's a model drug, this FCCP, and you see, and this is what you expect with the oxygen consumption rate is to go up. This is what you see here. This is published from microtiter plate experiments. And you see here, this is before poisoning, and this is after 10 minutes and after increasing time, 260 minutes. And you see also, we expose them subsequently to different concentrations of this FCCP. And you see also the influence of the concentration at 0.5. It takes 0.5 micromolar, it takes longer. And at 5 micromolar, it is faster, the response of this oxygen consumption. And at 20 micromolar, I think it is already getting toxic for the cells. That's why you have the influence on this negative influence on the consumption rate. And this is also the same is valid for the extracellular acidification rate. Also, you have here immediately after a few minutes, you see that the acidification rate is increased, and with 20 micromolar, it's already a high concentration for the cells to tolerate. So what we then did, we did also, and maybe this is especially important maybe for you, we did nanotoxicity tests with dynamic cell cultures. So, and this is what the experiment looks like. So we have here, this is the microfluidic chip, the cultured cells. So we use CACO2 and UVEC cells, and we have the integrated sensors, pH and oxygen, and also the fibers. You can see the chip here in reality. And we seeded the cells and then proliferate overnight. And then we expose them with nanoparticles really dynamically. And if you make these toxicity tests in static cell cultures, you always have the problem that the nanoparticles sediment and they sink down. And then, but with this perfusion, you mimic really real more physiological conditions. So, and with that, I've also picked up a few of these results. So here for the extracellular acidification with the silicium dioxide, nanoparticles with 25 nanometers, you see the light orange bar this is the acidification of the cells, and if you increase the concentration, you decrease the metabolic rate, which means you increase the pH, of course, because not so much CO2 is exposed, is released. Okay, and the really nice thing is we compared this with the conventional cell viability tests, and you see this hatched green is alive dead staining, and the green and the orange is the oxygen consumption and acidification rate. for Kaku and Hübeck cells, and also different concentration of the silica nanoparticles. And you really see that this is really comparable results. And with the difference that we really can do this sequentially, and it's not an endpoint. We don't use live dead stains. So, and then I have the next poll, and the next poll is, it's a simple question. So I would like to know if you know the seahorse system, and if people are familiar with that. So I see there's a quite high percentage of people who have who know it or have been working with that. So you see, this is a microtiter plate static cell culture system where you have these pillars here where the sensors are located, and with that you can also make these respiration measurements. And we compare this with a dynamic system, with a dynamic cell culture system. You see our microfluidic chip within this placed in the incubator, and so we will run the same assay that we do on the seahorse, and we run in this microfluidic chip. And the microfluidic chip, you see here a photograph of that, and the microfluidic chip has 4 integrated sensors, 1 pH and 3 oxygen per each cell culture, because we just cell culture here in one straight chamber, just to make it simpler. And we read that out with a readout system where we can have 48 fibers installed. That was a prototype of PyroScience. So, and then that's what it looks like in reality. Then you also have a lot of pumps, and here are the fibers and connected, and we could run these 8 channels simultaneously. And what we see then is when we do these standard seahorse tests, which one is the mitochondrial stress test, and you see that usually you measure the basal consumption, then you add some oligomycin to get the ATP production, and the FCCP to get the maximal respiration capacity, and then rotenone and antimycin will really block everything, and then you get the base line again. So, and that's what you see in the chip. So that is the measurement in the chip, and this is the measurement in the standard in the seahorse system. And you see they're comparable. And if you really calculate the numbers, you also see that they are identical, or in some cases the chip has even a higher respiration rate. And we also and then we also use the glycolysis test, and there is the same result. So it is also here we have this increase at the beginning, and then also the next step, the next increase, and this is also comparable to this seahorse standard. So, and what we see, so we compared now a static cell culture with a dynamic cell culture, and we see also that we have comparable results. I think that was important to show that this is because the seahorse system is a very established system, as we already also see in the poll that a lot of people know. Yes, and then I want to just quickly, because it's a really nice and fancy experiment, I think, or chip. We have this. I have been working together with Peter Loski's group, the Microorgano Lab, and he made this hard-on chip. So this is such a dog bone-shaped chip where he fuses spheroids from cardiomyocytes, from iPSC cardiomyocytes, and then they form contracting cardiac muscle fibers. And they integrated their electrodes, and we integrated in an oxygen sensor. And this is what it looks like. So this is a 3-fold chip with a membrane in between, where then this cardiomyocytes, muscle cells are forming. And you see, this is the oxygen sensor. This is a sensor which is about 300 micrometer in diameter, and we read this out from below. And the nice thing, if we now paste the electrons, so we have this. We stimulate the cells, they start to contract, and we have pacing frequencies indicated here, and you see here then immediately when the pacing starts, the oxygen consumption increases, and which is reflected in the oxygen concentration of the dissolved oxygen. And when the you see it here also in the second chamber, and when the frequency is increasing, there's also be more oxygen consumption. What you see here on the right side is just to confirm that this is not oxygen that is generated by hydrolysis. Okay, and then I come to the conclusion, and you see, I think we demonstrated inline measurements of metabolic parameters and bioenergetics, and this is an additional feature of these sensors to this monitoring on control of the culture conditions. With this, we can monitor this metabolism of the cells, and we have really short analysis times. It's only 2 minutes when we stop the flow, and then we analyze from these. And it is label-free, so it's non-invasive, and we don't have endpoint assays, so we can repeat the measurements. And we can it's a flexible integration into various chip designs. You see that, I think we integrated in this hard-on chip. We have also more simple microfluidic chip. We have also already some other more complex, but yeah. And we have a measurement platform for several parameters. I'm working also on gluctate and lactate, and also temperature. And the whole technology can be easy integrated in the chip production process, because we produce it from polymer formulations. And my vision is that I have a user-friendly, out-of-the-box disposable that is pre-sterilized, that the researchers just take out, and then they really can work with it. And I think the technology has really the potential for that. And maybe you know that also from microtiter plates or other labware where it's pre-sterilized and you just use it like that. And it is relatively easy technology transfer possible, and we are already doing that with company PyroScience. So, and then it's almost on my end. I want to say thank you to the people who contributed. So it's my group at the Institute of Analytical Chemistry, then Peter Ertl group in Wien, Peter Loskil's group in Uni Tübingen, and the Leiden University Medical Center with the group around Christine Mammery. And I also want to say thank you, of course, to the Austrian funding agency and the European Union. And you for your attention. Thank you.
Then we will directly move on to stay within the topic of in situ monitoring, but this time more on the imaging side. So what our group at the University of Tübingen is interested in is the broad field of regenerative medicine. And what maybe the one of the other here in the audience is also working in the field, but even though tissue engineering approaches and regenerative therapies and medicines is not a super new field, it has been out there for several decades, we still don't see that many applications that are actually out there existing in the clinic in the area of regenerative medicine. Some bottlenecks that we encounter here is that the understanding of the cellular mechanisms are so far not that well understood, because it often occurs that in a 2D or even in an animal model, our product or system or tissue aggregate seems to work fine, but then the issues occur at later stages. And some of the bottlenecks are identified here. So to better understand the cellular interaction and especially dynamic cellular processes, we need to get a better knowledge of, for example, cell material interactions. We need to have better test systems that allow us to also monitor these processes in vitro, and that we also have quality control tools, ideally even capable for in vivo follow-up. So we discussed this already in the first talk, to monitor 3D cell cultures, and we also saw that in the poll. There is some conventional readouts which can be either direct, so most of you said immunofluorescence is something you often use, but also gene or protein expressions, indirect by assessing in plate-based assays, for example, secreted molecules. But as Torsten already explained, they do have certain limitations, which is the endpoint, which is often a destructive sample preparation, but which can also be the spatial resolution. So often those assays just allow for a batch analysis. And this is why our group is trying to complement those conventional readouts with non-invasive imaging. So the aim here is to use intrinsic biomarkers, so biomarkers that already exist in the sample, which do not require the application of external dyes. So we also can circumvent pre-processing or staining procedures, which allows us for in situ measurements. And ideally also we are interested then in identifying novel biomarkers. The major techniques that we are working on is Raman microspectroscopy. I just saw a very few hands up for spectroscopic techniques in the polls and the methods you use. I will quickly elaborate on that. And then the other one, which is fluorescence lifetime short film microscopy. Both are non-invasive as they work based on the interactions between the photons of a laser and the molecules of our sample. On the Raman, which is shown here on the left side, we do have changes in energy due to vibrational stages of the molecules, which is detected in a spectrograph, which represents as a molecular fingerprint, which we also then allows us to have a specific Raman signature for every molecule, similar to a human fingerprint. These can be either as single point measurements, but we can also scan a certain sample size or area, and each of the pixel then provides that spectral information and allows us, by assigning pixel to similar spectral signatures, we can generate false color-coded images. Then we have limb microspectroscopy. The physical principle here is something that you might know from basic fluorescence microscopy, but instead of looking at the intensity of a fluorophore, we are rather looking at the lifetime of a fluorophore. So how long a fluorophore is bright. This is also something that is involved in the sensor techniques, for example, that Torsten just mentioned. And we can use that for single point measurements again, but what we apply this for is, again, imaging in a video rate quality, and it can be used on fluorescently labeled dyes, which, for example, then are sensitive to oxygen or temperature. However, there's also certain intrinsic endogenous molecules in our cells. So, for example, NADH and FAD, which are involved in the metabolism of the cell, which allow us to work marker independent in that imaging. Here is an example how such images could look like, for example, on in situ measured 3D cultures. On the left panel are some examples of Raman images. One focus area we have in our lab is to look at personalized medicine approaches, so patient-derived, either tumor organoids or tissue spheroids, where here you do see different colors, which do not occur due to any dye, but really just occur based on different spectral fingerprints and can, for example, be assigned here in the middle one to blue, here demonstrated based as nucleic acids, proteins, so different subcellular structures can be identified. On the left, this is how fluorescence or limb images can look like. The timeline you see here is a drug treatment or different drug treatments of, again, cancer, tumor organoids, where then those lifetimes changes due to different metabolic processes that go on. And in this false color-coded images, we do see that effect immediately. I now just have one example to bring you that whole principle of non-invasive imaging techniques and maybe also the advantages, and maybe all the limitations that come along with it here in a pancreas on a chip model that we did together with the microphysiological lab in Tübingen from Peter Laskill. So the idea here is that in our group, we were interested in producing insulin producing cells as a potential transplant, looking like here as an injection to encounter diabetes in diabetes type 1 patients. However, before, of course, implanting these cellular aggregates that are supposed to react to glucose by secreting insulin into a patient, we wanted to make sure that they are actually functional. So for this, the microorganal lab designed a microfluidic chip platform for us, which allowed us to track those beta cell spheroids in a certain space and allow perfusion. So by that, we could have living beta cell spheroids, which could be then perfused with different nutrient solutions, for example, but could also be applied for pharmacological testing. And then using that system, we wanted to perform quality control with non-invasive imaging techniques. So what we first did on that platform was Raman imaging. This is how those Raman images look like. Again, those color-coded images here are generated based on different spectral signatures here. On the example, for example, we have lipids shown in green, mitochondria or mitochondrial activity shown in pink, and nuclei due to those spectral fingerprints. They were perfused with either a low glucose, here called Krebs solution, or a high glucose solution. And then we do have different options to analyze those images. So the one is quantitative, as you are maybe used to analyze your fluorescence images. So you can just use those images because the signal intensity or the gray color-coded heat map correlates to the signal intensity of that certain molecules. So this allows us for quantitative analysis, where we could show that upon glucose perfusion, the mitochondrial activity or the metabolic activity was increased in the beta cell spheroids. For the other components, we did not see quantitative difference. However, we can now go a step further because in every of these pixels, we actually still do have that underlying spectral information behind. So everything which is now here, for example, green, represents the overall entity of lipids. However, there could still be compositional differences in the lipids. And to investigate that, we extracted the spectra. What is then usually needed to identify those smaller spectral changes is multivariate data analysis, so that we break down these spectral features. And what was shown that even though there was no quantitative differences in the lipids, there were qualitative differences in the lipid composition. And we were even able, by identifying the peaks that were different in the lipids, to assign that to different phospholipids. And the type of phospholipids which we found that were accumulated in the glucose-stimulated spheroids were assigned to insulin transporting vesicles, which does make sense. The same platform could then also be used to do fluorescence imaging. I do not want to go too much into detail on what kind of parameters are generated here, but I already said that it is based on these enzymes, NADH and FID, which are involved in cellular metabolism, and they can have, depending on their environment, shorter or longer lifetimes, and also depending on if they are bound to the mitochondrial membrane or cytosolic. And some potential readouts that can be made on these parameters to allow metabolic monitoring is, for example, a shift from glycolysis towards oxidative phosphorylation, so something similar, which you already saw previously on the sensor presentation, and which is also done in the seahorse assay, but which can now be assigned to an actual image. So, for example, here cell death can be monitored. Another parameter that can be accessed is oxidative stress, for example, caused by oxidative species, or a shift between aerobic to anaerobic metabolic pathways. Here in that image, you see the same organoid that could be monitored alive in that platform because it was constantly perfused, but it underwent hypoxia. So it was perfused, but under hypoxic conditions. And we could monitor that spheroid over a certain timeline. And we do see an increase in the lifetime by, or defined as a change in the color of the images. And the organoids are still completely alive. So this, in the end, for example, would be a potential application that before we would want to further use those spheroids, we could use them for screening to, for example, discriminate hypoxia damage from healthy or functional organoids that can then be used for an application or which can be sorted out and discarded. And we can do that on the same spheroid that we still want to use furthermore because we are not harming the spheroid. So in conclusion, I hope I could, or I hope you agree with me that non-destructive imaging technique provide insight into dynamic cell culture processes so we can monitor not just metabolic activity, but for example, also tissue remodeling or access decomposition of a tissue. What we also could show is that the Raman signatures differ between different cell types, so also cellular phenotyping, especially in the immune cell type field, might be of interest. What is also maybe, and compared to other microscopic techniques, interesting is that we, depending on the laser we use, we have some sort of penetration depth. So what you see on the right in that image is a bioprinted ink containing cells. So the cell signature is in blue, and we could scan through that whole bio ink for at least 1 millimeter in depth, which is a way broader penetration depth than confocal microscopes can reach. And we cannot only measure the cells which are in our system. In theory, also measuring the surrounding medium would be possible. So for example, also metabolites, as shown here in the right panels, like glucose or lactate, do change linearly in their intensity based on the concentration in the medium. So in the future, those techniques provide complementary readouts, especially suitable for in vitro test systems. Here is, for example, an organ-on-chip system displayed where we do have several access points where we actually can use those techniques to monitor certain processes because they are non-destructive and allow for temporal as well as spatial resolution. All right, with this, I hope I could also give you a short insight into the non-invasive imaging side. I think our group in Tübingen and also my team in Reutlingen, the microorganal lab in Tübingen as well, and then our collaborators from the University Hospital and the hospital in Stuttgart, as well as also the 3R network for inviting me here to give this talk. Thank you.
All right. Thank you so much for these two amazing talks. And we already have some questions, and I just invite everyone to keep asking questions, submit them through the Q&A button down below in your Zoom window. And the first one that came in from Sylvia Barker is actually, we have more questions. It's not just one. So I will just read out the first one. She says, great talk. I can only say that too. And then the question is, how stable are your sensors? So this is a question that goes to Torsten.
Yes, okay. And I will also show that on the screen again. You see this is the stability of an oxygen sensor spot to 200 micrometer, very small one. And you see there over 50,000 measurement points. So I would say there's no drift visible. And also this is the pH sensor. So we stored that over 9 days, and you see how the calibration curve is there influenced by that. I think this answers this question about the stability. And for different sensors, yeah, we have the same system. We always use this red light excitation. I can show you. I have a spectra here. I think you see that's what is. So we have this here. We have this. This is the benzoporphyrines. So this is the oxygen indicator, and this is the pH indicator that we're using. And you see here they can all be excited at 620 nanometers. This is where we excite these, and this is where we take the emission above 700 nanometer. That's what we detect. So yeah, and that's why we and on this dice we build up the platform, also the glucose sensor. So that's an enzymatic sensor that measures the oxygen consumption, and so that's our platform that we're using. And the nice thing about this is that we use red light is that it's less harmful to biological material. We suppress the background fluorescence of the biological material also, because if we excite with red light, there is no real background fluorescence. And we don't interfere with blue and green and red fluorescent stains. Yeah, so that's I think that answers also the question. Limited detection is a little bit difficult to answer. Maybe you've seen, when I go back to my presentation, we only measured 2 minutes from what you have seen this response, because we used only 2 minutes. So I think the pH sensor is 0.01 pH units, the resolution, and also for the oxygen, it's a 1 hectopascal or something. So so that's, I think that I hope it fully answered your questions.
Indeed, maybe I also have a question also for you, Julia. With the microscopy, you have also phototoxicity. I mean, it's something that one should be careful with.
Yes, of course. I mean, it depends on which laser you use and which laser power. Usually we have quite low laser powers. But that has also been defined what is useful and what is not. The good thing is that usually if they are in an aqueous environment, which is usually the case in 3D culture, because we do have the medium surrounding it, then that heat effect, which can occur due to a laser, is often buffered. But so far for the applications and the integrations times that are used, it is not a problem for the cells. But of course, if you go full power, you can also burn your sample.
Thank you.
Maybe adding up to this, Julia, how long does one of these measurements take?
Yeah, so the film is quite short. This is comparable to fluorescence imaging. So depending on the resolution and the area you aim for, it's done in 1 minute to 2 minutes. The Raman takes longer because we acquire a spectrum in every pixel. We can tailor our resolution and our image frame if we want to go for a larger tissue, but don't need a super tight spatial resolution. We can go down in that. Raman originally comes from material sciences, where they are looking at atoms. Then you have a very small frame, but want a very high resolution. But basically saying a measurement of the single cell takes like 2 to 3 minutes, but of a tissue section of maybe 200 to 200 microns is more like half an hour to an hour.
Okay, thank you so much. Just checking if new questions came in. Right now, this does not see. Yeah, here is another one. It's also for Julia. The question is, can you image tissues?
Yes. So tissue sections is quite easy. It can be done on histological sections, for example. There's also something we do, for example, tumor margin detection. In vivo applications are also possible. There is first studies on clinical applications of using Raman spectroscopy, for example, in an endoscopic setup. But there, of course, again, the time is an issue. But in principle, yes, also living tissue is feasible.
Okay, thank you. Now, maybe a question to Torsten again. We talked about it, like different methods now. So do you see, is there any way to combine these methods to maybe do them all together at the same time?
Yes, I think that is possible because we have these sensor spots. They can be put somewhere in the chip, and then we can measure that. And I don't think it interferes. The light beam is probably interfering, but I think it is doable. It's possible if you arrange it. And if you think of how to arrange it, it is possible. I don't see a problem in that.
Okay, there is a question in the chat for Julia, if you can retrieve the spheroids after imaging.
In principle, yes. So I think, if I understand the question correctly, it is about if we still can use the spheroids after imaging. Yes, for both Lim and Raman, as they are not interfering with the cellular properties, you can. And you could also do a time series. So you can either keep them at the microscope stage and do an acquisition every half an hour, for example, or put them back into the incubator, get them back 3 days later and image them again, if that was the question.
I have a question on how these spheroids are trapped into the chip. I'm not so familiar. It's just nice, this animation, but how does it really work?
This is microfluidics, so you have to ask Peter in detail, but based on the flow, they do simulations. And of course, they do have to have a certain size. The flow rate also has to be a certain flow rate, because if the organoid is too small, it might flush or try to evade through that little hole. So it really has to be designed based on the size of the spheroid and based on the flow that is applied. But if they are in balance, then the flow is just enough that the cells start point by point. You really can they are lining up one after the other in that microfluidical system. And then if there's a constant flow, they are in there. If you release the flow, then they can start moving around again. So it's really dependent on the flow.
Quite cool.
We have another question for Torsten. So the question is, can any surface be functionalized with your sensors, or do you require a specific material? It is adherence can be a problem, and we find solutions with primers, for example, if it's not adhering. So we have oxygen sensors. We have immobilized now on glass. We have another sensor formulation than for plastic, like COC, so there's different possibilities. With pH, I'm at the moment, I have first tried, I've solely tried on plastic COC with a rough surface, but we're also working on that. This is going to be more flexible then. Yeah. With glass, this can be difficult because then we have a hydrophilic pH sensor material, and on this glass, the adherence, but we also have already found ways to do that. But I think you have to put some effort in, but it's usually it works. Yeah. Maybe adding on to this question, if we talk about the surface of the materials, how is it with the sensor materials themselves? So did you look into if there's any other, let's say, interaction or interference between the cells and the sensor materials directly?
Yes, we have also, so what we use is for oxygen sensors. It is also polystyrene, which is used for cell culture materials. So that's we made their sensor spots, and they really grow around this because they don't like that hydrophobic surface. But we also have another material where you use hydrogel that is FDA approved. So I don't see there any interferences or toxicity from that. And the dyes are incorporated in a really, really low concentration. Yeah. So if there's some released, then I don't suspect there will be anything. And we have already made some cytotoxicity tests in other projects. Yeah.
Okay, thank you. Maybe just adding on to this materials question. So question for Julia, how is it with the, for example, the Raman spectroscopy in terms of materials? Are there any limitations? For example, if you think of cell culture dishes or dishes where your organoids or whatever are in?
Yes, it actually is. That's the thing about Raman. It is kind of like generating a fingerprint from every molecule. And unfortunately, polystyrene, for example, or plastic, has way higher Raman signatures than biological signatures. So what for sure is not possible to penetrate through a plastic which covers your sample. And what we also usually use is glass bottom dishes, and also, for example, for those microfluidic platforms, PDMS can be an issue if it's too close to the cell layer. So there needs to be an optically inert material, which usually is glass. It doesn't have a big Raman signature, or for example, calcium fluoride. And then it's possible, but yes, there is some restriction in the selection of the surrounding substrates.
Okay, thank you very much.
There is a question, maybe the last, I think, unfortunately, for Torsten, is can you recapitulate zonation gradients? For instance, in the liver sinusoid or intestinal villi crypts with your sensor, for example, by serial connection?
Yes, you mean that what you maybe think you can do is to have several spots in there. Yeah, and then you really can measure at several, if you have several measurement points. That would be a way to analyze that. On the other hand, there is also the possibility to do lifetime imaging. So it's more fluorescence lifetime imaging if you have the right setup for that. This could be also, we have something like that in the lab, such a camera. And so then you really see the gradient visualized. Yeah, so that's possible. Yeah.
Okay, thank you. And yeah, I think we're a bit running out of time. So I would like to thank both you and Torsten for the discussion and replying all our questions. And maybe I will just share very quickly information for the next appointment of our webinar. Yeah, so we will have a video you see last Thursday. So the next one February with Katrin Hermann on the
we are losing Georgia, but I can maybe take over. Georgia, your connection broke down, so this is why I don't know if you can hear us right now. But the next webinars are going to be the one in February. We will have Katrin Hermann, from the Landestierschutzbeauftragte in Berlin. And then in March, we have another talk on this time in silico methods, which also will be really interesting, I think. You can also check them out on our website. There we also announce the new webinars. So if you don't have a chance, and now you can actually see it again. And then also, as Georgia mentioned in the beginning, if you want to rewatch one of our recordings, then just go to our YouTube channel or again to our website. And there you can find all recorded webinars and you can watch them and share them. And we really hope to see you all again in February. As always, it's going to be the last Thursday of the month at 5. And thanks again to our experts today, Julia, Marzi, and Torsten Mayr. Also, thank you, Georgia. And we wish you a nice evening and hope to see you soon in February, or then also hopefully in Berlin. So I posted something in Georgia, also can show it again. So if these topics are of interest to you, check out the website for the MPS World Summit in Berlin this year in June. And I'm pretty sure we will see a lot of faces from today again in Berlin. All right. And with that, thank you to the audience and to all of our participants, and again to our speakers, and have a nice evening.
Okay.
Goodbye.
Bye-bye.
Hey, goodbye.
Bye.
Bye, everybody.